So as we move into our next unit, we're going to start talking about how cells divide, going through a process in order to take one cell and turn it into two identical copies. This process is known generally as the cell cycle when we talk about eukaryotic cells. The process used by prokaryotic cells is called binary fission. Now this is an important process for prokaryotes to reproduce. One becomes two. In some cases that'll only take 20 minutes, and it allows their population to grow rather quickly. So consider food poisoning. If you've ever gotten food poisoning, you ingested a bunch of bacteria, they replicated, and within sometimes less than an hour or maybe 2 hours, you're suffering the effects of food poisoning or a large increase in a bacterial population in your body. But when we look at eukaryotic cells, the drive behind cells dividing is to make an exact copy. This is what eukaryotic organisms like humans will use for growth, repair of an injury, development of our body, our hair gets longer, etc. All these things are due to cell division. So we're going to explore the process and look at how cells divide, what are the steps, the requirements, the mechanisms necessary for cells to become two cells. And then those two divide to become four, 4 to 8, and so on. And we want to keep in mind, how long does it take? What structures are necessary? What ingredients have to be there? And what's going to happen if something doesn't go correctly? What are the potential consequences? So with prokaryotic cells, they use a process known as binary fission. Binary fission involves 5 key steps. So it's somewhat of a 5-step process. So during the first step of binary fission, the DNA of the prokaryotic cell will replicate. So that blue loop of DNA copies itself. Hopefully everything is done correctly, and the original code of DNA has now doubled and created a second identical code of DNA. The cell then begins to elongate, and it will stretch itself out. As it elongates during the second step, it's starting to pull the DNA loops apart. As it moves to the third step, the DNA loops will actually split and get pulled to opposite sides of the cell. That way one loop goes to the right side, one loop goes to the left side, and the DNA is physically separated from each other. And then as it moves into step 4, a dividing wall or line or structure called the septum will begin to form. So this is how the cell is going to actually get split in half. So it elongated into this rectangular or tubular structure, and the septum begins to split in half, ideally in the center of the cell, so that one loop of DNA is on the right, one loop of DNA is on the left. As the septum forms, it then moves towards step 5, which eventually pinches the cell completely in half, and you have 2 bacterial cells sitting together side by side, each containing what should be an identical code of DNA. Now, in some bacterial types, this is an incredibly fast process. E. coli, 20 minutes. So 20 minutes to go from 1 to 2, 2 to 4, 4 to 8, and so on. It's unbelievable, the replication rate of bacteria. One cell can divide into 10 billion cells over the course of 10 hours. So when you look at growth in a bacterial population, it's staggering because of this rapid process of binary fission. Now, ideally, every single cell is identical, but the reality of replicating your DNA that many times in such a short period of time, such a condensed amount of time to take the DNA, replicate it, and split the cell in half, it significantly increases the likelihood that mistakes will be made. And when mistakes are made, that sometimes can lead to new genetic variations. So this is why we get antibiotic resistance evolving in bacteria because it replicates very, very rapidly. And when we hit it with an antibiotic, we're wiping out certain bacterial types, but because of replication and mutation and new variations, some of those things are not wiped out by the antibiotic. So consider the amount of antibiotic-resistant bacteria we have in the medical system today. I mean, you have MRSA, which is resistant. You have tuberculosis, which is resistant. You have all sorts of different bacteria that are resistant because we've wiped out the weakest, left behind the strongest, and because they have that rapid reproduction rate of binary fission, they're able to reproduce very, very quickly. Now, when we look at other organisms, the cell division is going to vary. And there's a pretty big diversity of cell division among different organisms. So prokaryotic cells follow generally the same pattern of binary fission. When we get into different protists, this is a very unique, kind of goofy group of organisms. They have different ways of replicating. We get into fungi like yeast. They have a different method of replicating. And then the animals and the plants and other organisms have some slight twist on this variation of replication. Our main focus, though, is going to be looking at how animal cells replicate through cell division. What are the key structures? What makes it unique? And then we'll also look at plant cells. And we want to do a comparison. Similarities, differences between plants and animals. But do keep in mind there are a lot of different variations on cell division. But generally, the end goal, one cell divides into two, and they're supposed to be identical. It's the end goal of cell division. Okay, so as we look at cell division in the eukaryotic cells, the key thing to watch is the amount of genetic material, total number of chromosomes. So what do you have there in that cell? How many chromosomes? Those chromosomes have to be divided. They get divided. Then the cell has to make new chromosomes. So it's a number that we want to keep in mind as we look at cell division. So just a couple examples highlighted on the table here. Humans, we have 46 chromosomes. 23 pairs, or we say 46. Chimpanzees, they actually have more genetic chromosomes, more DNA than us. They have 48. Heck, you look at something like the sugarcane plant that gives us sugar, what we use for cooking and consumption. 80 chromosomes. That's a whole lot of chromosomes. Mosquitoes, though, pretty simple. 6 chromosomes. Drosophila, those fruit flies, only 8. So, things to keep in mind about chromosomes. It's not how big your genome is. It's not how many chromosomes you have that make you a better or worse species. It's what information is contained in that. So, typically humans would say, oh, well, we're superior to all these other species. The frog and the mouse and the possum and the fruit fly because we have more DNA than them. But we were mentioning earlier, chimpanzees, more chromosomes than us. Look at a chicken. Look at a dog. They have more chromosomes than us. But our DNA has specific genes that enable us to adapt and evolve to become what we are. Those other species, same thing. They have specific genes that enable them to adapt and evolve and become what they are. What we are learning, and we'll discuss this more in the biotechnology unit, we are learning that there are often very similar and sometimes identical genes between different species. So the dog has more DNA than us, the chimpanzee, the horse, the sugarcane, etc. But when we look at the information contained in those genes, we see an overlap. We see sometimes the identical gene found across multiple species. So again, something we'll discuss at a later point in more detail. Okay, so let's take a look at the eukaryotic chromosome. The eukaryotic chromosome should have 46 chromosomes. That's how much should be in, for humans, I should say, 46 chromosomes. If an organism is missing a chromosome, it only has 45 in the case of a human, it is considered monosomy, or you'll see the term monosomic. Mono means one instead of pair. So monosomic, you are missing a particular chromosome. Now this can happen on any of the pairs of chromosomes. Normally humans have 23 pairs or 46 total chromosomes. If for some reason one of your pairs of chromosomes is short, and instead of 2 chromosomes you only have 1, it would be monosomic. In most animal species, monosomy results in death of the organism very, very early on. A lot of times it doesn't even develop. It starts going through the developmental process, but there's a lack of information that the cell cannot overcome, and it does not develop and survive. When we look at an organism that might have actually an extra chromosome at a particular pair, then we call that trisomy. Tri refers to 3. 3 chromosomes at a given pair. The most common trisomic individuals are those we call trisomy 21. This is what we know as Down syndrome. Down syndrome individuals have an extra chromosome at a 21st pair. So depending upon the level of expression, it could be very minor. There could be no real noticeable effects of the Down syndrome, or the other side of the spectrum, it could be debilitating and unbelievably damaging and cause significant problems for the individual. And there's everything in between. But trisomy, or you'll see it called trisomic, refers to 3 chromosomes at a specific location or pair. Some species, trisomy actually is a good thing. In certain plants, trisomy enables them to do additional things. So it's not always a bad thing. But for animals, it does tend to be a negative scenario. Okay, so inside the nucleus, before we said we have 6 feet of DNA, and that DNA is a big pile of loops of DNA called chromatin. That chromatin is roughly 40% DNA, actual coded information, and then 60% of it is made up of proteins, little protein structures that help hold and act as anchors in organizing of the DNA. So 2 terms to be familiar with. They're bold, and I left them in there. The first term is called heterochromatin. This is part of your chromatin, or your DNA, that does not get expressed. It does not turn into anything. As far as we know, scientists have not discovered really what is the reason or use of that information. Euchromatin is the pieces of information, or chromatin, that actually codes for a specific protein, which then becomes expressed as a specific trait. So your hair color, your eye color, your height, your weight, your blood type, all these features and traits about us that are expressed are due to the information contained in the euchromatin part of our DNA. So in general, when we look at DNA, we have 6 feet of it. We will use only maybe 1 to 2% of our genetic code for expression during our lives. So there's a whole bunch of heterochromatin that we never, ever use. Some great research is happening right now looking at what does that do? Is it actually more important? Is that a regulatory piece of information that actually influences the expression of the traits? So more research is needed. We'll hopefully know in the future.